Sorry. Hereby I open this academic ceremony in which Ms. Fatma Mokhtar will defend her academic thesis entitled Intestinal Cholesterol Absorption in Humans, Focus on Genetic Variation and Molecular Pathways. May I invite you, Mrs. Candidate, to present a summary of your studies and the conclusion of your thesis in the next 15 minutes. Thank you.
Thank you. Dear Pro-Rector, Assessment Committee, my supervisors, family, friends, and colleagues. In the upcoming 15 minutes, I would like to present my thesis titled Intestinal Cholesterol Absorption in Humans, Focus on Genetic Variation and Molecular Pathways. It's known that high cholesterol levels can accumulate in the blood vessels, which can cause plaques. These plaques can reduce the blood flow. This can increase the risk of developing heart attacks and also strokes. Cholesterol is mainly synthesized in the liver, and it can also be absorbed via the diet that we eat, via the small intestine. There should be a balance between cholesterol synthesis and absorption to maintain overall health. What are single nucleotide polymorphisms, SNPs? The DNA consists of 4 letters, A, T, G, and C. And as we can see here, we have 2 individuals, 1 and 2. And we can see that the sequence is the same of those 2 people, except at the position, the T, the thymine, is replaced with a G, a guanine. And by this, we have a SNP. So it's a change in the nucleotide base at the same position. And that we name a SNP. The SNPs can have an effect on different traits and also in the risk of having a specific disease. It has also been found that the level of cholesterol absorption varies widely between individuals that have taken a standard meal. This variation can be due to genetic variation in the DNA. Moreover, there were different diseases that were associated with the level of cholesterol absorption. Those people with high cholesterol absorption, they have a higher risk to develop cardiovascular disease, while people with low cholesterol absorption have a higher risk to develop steatosis, which is a fatty liver disease. Why is it so important to categorize individuals as low or high cholesterol absorbers? If we can do that, then precision medicine will be a much easier way to intervene. So you can give the right medication to the right patient. How to measure intestinal cholesterol absorption? Normally you can use isotopic tracers. These are radioactive compounds that can measure the cholesterol absorption. But that is a very labor and costly method to do. Instead, we can use the blood. In the blood, there are markers that reflect the cholesterol absorption. We have the campisterol and cetosterols. And for endogenous cholesterol synthesis, we have the latosterol. All these markers are validated markers. What was the aim of this study? We wanted to test the association of genetic variation in a European population with the level of absorbed cholesterol. And we were also interested in creating an intestinal cholesterol absorption network. Moreover, we wanted to categorize individuals as high cholesterol and low cholesterol absorbers based on their genetic profile. First, we have performed a systematic review. So we have looked into the literature and we have selected all studies that have tested association between SNPs and intestinal cholesterol absorption rate. After filtering the papers and reading the papers, we have found that variation in 7 genes were associated or related with the level of cholesterol absorption. And as we were interested in creating the intestinal cholesterol absorption network, we have used these 7 genes in creating the network. So as you can see here, the circles in black are the genes of the literature. And the circles in gray, these are computer-generated genes. And the bigger the size of the circle, the more likely that the gene is related to the network. In total, we have found 15 new genes. Then in chapter 3 and chapter 4, we have used data of 471 participants. They were all European, and their genetic variation was read using a precision medicine research array. By this array, you can read the genetic variation in the DNA. And 3 parameters were available for this population. So intestinal cholesterol absorption markers, the endogenous cholesterol synthesis, and the serum low density lipoprotein cholesterol. So in the first study, we have pre-selected some SNPs that were of our interest. So we have selected 6 genes that are responsible in endogenous cholesterol synthesis and 3 genes that are responsible for intestinal cholesterol absorption. And we have tested the genetic variation in all those 9 genes with the available parameters, so with cholesterol absorption, cholesterol synthesis, and LDL cholesterol. What did we find? Here we have in this table, the ABCG5 and ABCG8. These genes are responsible for the efflux of cholesterol from the anterior side to the out of the cell. And the variation in these genes were associated with the level of cholesterol absorption. And for the MPC1L1, that gene is responsible for the influx of cholesterol from outside the cell to the inside of the cell, and the variation in these genes were associated with endogenous cholesterol synthesis. Then we have also tested association of, we had 2 genes in the liver that are responsible for the endogenous cholesterol synthesis. Variation in these genes were associated with LDL cholesterol levels. Then in the next study, we have decided to test all the variation that were available. So we had 160,000 SNPs available, and we have tested their association with both intestinal cholesterol absorption, campisterol and cetosterol. What did we find? Regarding the campisterol markers, we have found many SNPs that were associated with campisterol. That is a Manhattan plot that is presented here in this figure. On the x-axis, we have the chromosome. We have 22 chromosomes. And each chromosome have a different color. So as you can see, number 1 at number 1, that represents chromosome 1. And all the dots in black are the variation on this chromosome. And the next one is in gray, so all variation on the second chromosome are represented in gray. And on the y-axis, we have the minus log p values. And the blue line indicate the threshold of significance. So all the SNP that reach a significant association with campisterol are presented upon this blue line. Then again, we did the same. So we tested the association of all the SNPs with cetosterol. And again, we have different SNPs that reach it out, and they reach a significant association with cetosterol. So as we have tested the association with both markers, only SNPs that showed consistent association for both markers, so for campisterol and cetosterol, were considered to be significant and relevant. 14 unique genes were expressed in the small intestine. Yeah. Then in the following chapter, we had another study. We had data of 20 participants. The first part of these participants have taken plantastanol ester. That is a compound that can reduce cholesterol absorption. The other group was the control group. So this group of people, they did not receive this compound. When the participants arrived to the university, first blood samples were collected. Then they have received the shake. The shake contains plantastanol ester that reduces the cholesterol absorption. After 4 hours, blood was collected again. And then after 5 hours, biopsies were taken from 2 parts from the small intestine, from the first 2 parts, from the duodenum and from the jejunum. And the reason for that was to read the gene expression of those 2 parts. We had around 9,000 genes for each part. After a period of time, the participants have been switched to the other group. So those who have taken plantastenol ester, they were switched to the control, and those who were in the control were switched to plantastenol esters, and the procedure was taken again. So for each participant, we have blood samples, the campestyrol markers, and we have their gene profile before and after the intake of plantastenol ester. As we were interested in categorizing those individuals as a high and low absorber, we have used the campestyrol's total cholesterol standardized campestyrol. We have ranked those people from the smallest to the larger, and those with the lowest concentration were categorized as low cholesterol absorbers. We have taken only the 3 lowest. And the highest, with the highest total cholesterol standardized campestyrol, were categorized as the high cholesterol absorbers. Then we have checked for their gene expression. So as we can see here, we have the Venn diagram. First, we present here the duodenum part. We can see in the green circles that the low cholesterol absorbers had 482 differently expressed genes, while for the high cholesterol absorbers, there were 181 genes differently expressed, and only 16 genes overlapped between those 2 groups. And in the duodenum, again, we see that the high cholesterol absorbers had 366 differently expressed genes, and in the low absorbers, there were 316 differently expressed genes, and only 20 genes overlapped between those 2 groups. By this, we can say that the genetic profile after the intake of plantastenol ester greatly influenced the gene regulation for those 2 different groups. Then we were interested in testing the regulation of these genes. Are those genes going up or going down after the intake of this plantastenol? So for each group, we have selected the first 25 most significant genes, and we have compared them to the other group. So as you can see here, if a gene goes down or downregulated, then it is indicated in blue, and if it goes up, then it is indicated in green. And we can see that most genes are going to the opposite direction. So if you see in the low absorbers, a gene goes down, then it goes up in the other group. After that, we have performed an enrichment analysis using different databases. One interesting pathway that results from this research was the anterior site cholesterol metabolism. So we have decided to investigate more in this pathway. It's known that the intake of plantastenol ester will reduce LDL cholesterol, but the reduction level is different between high and low absorbers. It's known that people with high cholesterol absorption have a more response to plantastenol ester after the intake of plantastenol. There is more reduction in LDL in the high cholesterol absorbers. So what is the reason behind this? Here, the black boxes represent the anterior site. We can see that in all the groups, except in the high absorbers in the duodenum, that intestinal cholesterol, endogenous cholesterol synthesis goes up. But in the duodenum, it goes down. And this may explain why those people benefit more from the plantastenol in lowering the LDL cholesterol. We have also looked into the MPC1L1. This is a gene that allows the entrance or the efflux of cholesterol from outside the cell to the cell. It goes down in the low absorbers in the duodenum, but in the high absorbers, it goes up. And in the high absorbers in the duodenum, it goes also down. Regarding the ABCG5G8, that is a gene that is responsible for the efflux of cholesterol, and we can see in all the groups that it goes down, which is also logical because the cell tried to compensate to the loss of the cholesterol that is caused by the plantastenol ester intake. Moreover, for the low absorbers in the duodenum, we see that ApoA1 also goes down. The ApoA1 is also an important gene that allow the efflux of cholesterol to the particles of high-density lipoprotein. And again, the cells tries to keep more cholesterol inside it. Finally, in the jejunum, the LDL receptor also goes up. So more cholesterol will enter the cell if there are more receptors on the surface. As a conclusion, new SNPs have been identified that are associated with intestinal cholesterol absorption. An intestinal cholesterol absorption network has been created with 50 new genes. 14 unique genes were associated with intestinal cholesterol absorption. The response and gene expression profile differed between subjects that were defined as high cholesterol or low cholesterol absorbers. Thank you for your attention. I give the word back to the prorector.
Thank you very much, Ms. Candidate. I think due to the time, we have to start swiftly with our opposition, and the opposition will be opened, of course, by the chair of your assessment committee, Professor Sverdlov. She is a professor in hepatic inflammation and metabolic health, and I would like to give her the first word.
Dear candidate, first I would like to thank you for a very clear presentation and also for your thesis book. I read it with a lot of interest. I believe that your work contributes a lot to our understanding regarding the tight link between cholesterol absorptions and genetics modifications, and as such can open the door in the future for precision dietary approaches, which is great. So I would like to start with asking questions about Chapter 5, and first about the experimental design. Actually, you started with 20 different individuals, and you performed the analysis to all individuals, at least you have the gene expression of all individuals, but yet you chose at the end to select only 3 per group. I wanted to understand what was the rationale behind it, and whether you think that it is sufficient actually taking into account the big variation in the population to focus just on 3 per group.
Highly esteemed opponent, thank you for your compliments and your question. Yes, in Chapter 5 we have selected, we had 20 participants, but we have selected the 3 highest and the 3 lowest. The reason behind this, we have ranked them from highest to lowest. And as we were interested in categorizing individuals as high absorbers and low absorbers, we did not have enough samples to include more people. And we did not include the participants that were in the middle, because our interest was more in those the highest and the lowest, because when you intervene or when you give specific medication, we know, for instance, as I have mentioned now in my presentation, that plant intestinal ester intake will influence or affect the people that are high absorbers in lowering the LDL compared to the low absorbers. So I agree that increasing the sample size and including more people can enhance our finding, but still we have found interesting results in gene expression in those 2 groups.
But you do have the data about the gene expression on all individuals, right?
Yeah, if we look into these numbers of the campus triol, we can see that the variation is somehow high of those, the highest, and in the middle they look similar. So we thought, we will only select. It is also possible to look in the middle group. So, but our interest was more focused on those 2 groups.
No, I was just confused because I think it makes a bit more sense or more practical if you would first decide who is high cholesterol absorption and who is low, and then do the analysis only for these patients, and you don't have to look also in the middle. But I understand. And do you think that within this number is sufficient to have conclusions that you can rely on, because of still there is a big variation and you have only 3 individuals?
Yeah, true. I would also say increasing the number of the sample size would be more, it gives more relevant results, but still it's a first study, and that was the data that was available, and I think we still see what we expected, that there is a difference in gene expression between the high and the low absorbers. It's possible in the future to redo this in a larger population and to see if we see the same results.
Okay. And when you talk about expected results, I was also wondering, because you took per individual's data from 2 parts of the intestines.
Yeah.
What was the aim there? Did you want to really show why would statins work better among, or what was the rationale there? What did you expect to see there? Was the results in line with your expectations?
Yeah, actually, it's still not known, and there's no many studies that have performed this kind of study. So we don't know how cholesterol absorption is still in the 2 parts. So is it more absorbed in the duodenum or in the jejunum, and how do statins or other drugs or plantastenol, for instance, affect cholesterol absorption? Is it that it works really on the duodenum or in the jejunum? So that is the reason for selecting those 2 first. And it's known that the duodenum and jejunum are the first part of the intestine, and that the cholesterol absorption is more absorbed in those 2 parts. So that's why we did it.
Okay, then it's very clear just to understand the mechanisms there, I understand. And about expected results, because you find an opposite effect, which I personally was surprised by that, because I would expect maybe to see the same effect, only to different magnitude, right? Because you'll say, well, stenols are more effective with high cholesterol absorption, then maybe I would expect the genes to go, or the expression of the genes to go to the same direction, but maybe to less magnitude. But you show totally opposite profile, so how is that possible? Because it's not so much in line with the clinical effect that we see.
Yeah, true. When we first did this analysis and we saw that the direction of each group goes to the other direction, so in the opposite direction, I think this gives a very clear indication that those 2 groups are so different. So if a gene goes up and in the other groups goes down, I think that indicates that the difference is very high in their response to this plantastenol ester intake. So we didn't look inside each gene, or we don't know the real function of each gene in each group. I think we still have to understand the function of each gene and what is the cause of going up or down.
But should we worry? Because, I mean, if it's beneficial for one group and it's opposite to the other individuals, that's maybe, I don't know, what could be the clinical applications of this? Can it shows that it can be maybe not good for some individuals to consume plant stenols?
Yeah, it's true. It could be that plantastenol is not efficient for every patient. As I have mentioned, those with high absorbers, plantastenol would be the optimal, or it is a good choice to use. But for those low absorbers, maybe we have to select another intervention. Yeah.
Okay. And do I have time for a very short question? No. No problems.
I'm very sorry for this.
Thank you very much. I'm very happy with your answers, and I bring back the word to the corrector.
Yeah,
thank you very much. The oppositional will be followed by Professor Mariman. He was also involved as member of the assessment committee, and he's professor in functional genetics at our university, and I'd like to give him the word.
Thank you, Mr. Prorector. The thesis. I found again that the cholesterol metabolism is so complex, but certainly you contributed with new insight and also provided leads for future research. I also want first to go to chapter 5 with you for some questions maybe, because you're interested in the difference between of the cholesterol metabolism between the low and high absorbers. But you do genomics, you compare them after the challenge with the control in a very nice crossover study. But would it be possible just to do a simple comparison of high versus low absorbers? So the data the gene data, not compared to the controls for each group, but compared to each other? Or would that give a different result?
Highly esteemed opponent, thank you for your compliments and your question. Yes, it is also an option to compare the gene expression of the high with the low, so not using the control group. And by this, I think you can gain more information about the two groups, for sure. In our study, the aim was to see the effect of stanols just in those two groups and to see. But if you approach this method, then you will check for the difference and you can get more a direct answer, I think, on genetic changes between those two groups.
Yeah. Okay. You find also, you do these enrichment analysis, so you have the pathways that come out. My first question would be, did you try to link your genetic results to the genomic results? That is, you find new genes, and are they included in these pathways?
Yeah. So we have performed enrichment analysis pathway by using 3 different databases, the KIG, Reactome, and Wiki pathways. And by this, we have used all the differently expressed genes in each part, so for the high, for the low absorbers in the duodenum and the jejunum. And for each group, we have performed the enrichment analysis. So, for instance, we have the anterior side cholesterol metabolism was a result of a Wiki pathway from the high absorbers, I think, in the duodenum. And all the genes that came from there are the genes that some of these genes have reached significant association, but some of them did not reach significant association as they were not a result of was not an input from our data. But it is the Wiki, for instance, this anterior side, we have some SNPs in the endogenous cholesterol synthesis that reach significant association, and it is included in this pathway. So for that reason, it pops out.
Yeah, that's clear for me.
Okay.
I mean, you also describe that several of these pathways do not provide new leads that can connect to our current understanding of intestinal cholesterol metabolism. So that sounds a little bit disappointing because you say, I do not find in these pathways new leads. Because they do not link to my knowledge of the cholesterol metabolism.
True.
But is that not very good? Are these pathways not giving you new information beyond the things that are already known?
Yeah.
You should go into that maybe.
Yeah, true. What we mean by this, we mean that our current knowledge is not sufficient to explain the link of these enriched pathways with intestinal cholesterol absorption. So we were also in our discussion, we did not speculate a lot about these pathways because we don't know the link of these pathways with absorption. So more studies are needed to understand the relation between them and intestinal cholesterol absorption.
Yeah, but I think they are good leads for future research, I think. Then about, it was already mentioned, but you can see it in the pathways of the low cholesterol absorbers. There are a lot of inflammatory pathways and cell cycle pathways. So I would think when I'm a low cholesterol absorber, I would better not take these stanol esters. What would you say? Because then some inflammation may start. Or was it due to the concentration of your challenge that you gave? That the inflammation pops up in your pathways.
Yeah, true. If the inflammation pathways pop out, it's not a negative thing, I think, because it gives an indication maybe that the low absorbers have a higher risk for inflammation in their duct. It's also known that people with low cholesterol absorption have a higher risk to develop, for instance, steatosis, this inflammation in the liver. that is the data that we get, but it also can, or we can speculate that those with low cholesterol absorption have a higher risk to get more inflammation, or that the inflammation pathways are involved, or they are more active in their body.
So you suggest that inflammation may be part of the cholesterol reaction, metabolism reaction?
Yeah.
Oh, okay.
It can influence the...
Yeah, for sure. I have to see. I looked through the genes, and my question would actually be, you have these tables of the 25 genes, huh? Did you actually go through the function of these genes to see if they are related to the cholesterol metabolism?
Honestly, we had 4 tables, and each contained 25 genes.
Yeah.
I looked randomly in some, but to be honest, we did not check one by one for the function. I would say because they are involved in, we have used these genes in the enrichment analysis, so it can give an indication of about the pathways that are involved in. But we did not look separately for each gene.
Yeah, because I was challenged or triggered a little bit by this SLC23A1, that is a transporter. In your low cholesterol absorbers, duodenum, you identify the 3 genes with the highest fold change, and that is one of them. And yeah, I would think then I will look into the function, but you didn't look further, is it?
Sorry, for which table?
It's on page 179.
Yeah.
There is SLC23A1, that is a transporter, and then if you talk about absorption, I think, oh, transporter, that's interesting. I'm going to look into the function. Did you do that, is my question.
No, honestly, I did not check for the function of...
Oh, okay. I'm satisfied with your answers and give the word back to the pro-rector.
Okay, thank you.
Thank you very much. The opposition will be now continued online. So we go to Professor Leitinen. She was also a member of your assessment committee, and she is professor in human nutrition at the Research Center for Integrative Physiology and Pharmacology at Turku University in Finland, and I would like to welcome her to join us this morning. I'd like to give you the word, Professor Leitinen.
Thank you very much and good morning. Yes, congratulations for the candidate and the rest of the team for the very nice and also challenging work. So I have some questions for you. It seems that chapter 5 is of interest for many of us in the assessment committee. I would like to ask a little bit about diet. You had here a stanol ester intervention, and you saw some differences in gene expression. Could one say that you had here kind of an acute response because you had the biopsies very soon after the intervention and so on? But if we think about how cholesterol metabolism is changed by lifestyle, that quite often takes a bit longer time, a couple of weeks or so. Did you record the diet in this study? And what do you think about the background diet effect on your findings?
Maybe you could repeat the question so that we know that you understand
Yes.
the question.
Did you record the dietary intake in the chapter 5 study at all, at any stage?
Highly esteemed opponent, thank you for your question. If I understand your question correctly, you are asking about the diet that the patients were taking during the study. Yes, what we know, first when the participant arrived to the university, they have taken first a lasagna to standardize their, that they have the same food intake. And but if you are asking about the period during the period that they have taken plantastanol ester and the washout period. The participants were only recommended to follow a specific pattern of food and to take, so those who have taken the plantastanol ester, they have taken 2 milligrams per day, and the other group, the control group, did not take this intervention of the plantastanol, and then there was a washout period of 2 weeks. But further, we don't have, as far as I know, we don't have data about the food intake.
Yes. Yes, so you had a standardized test meal, so that's excellent. You know what they have from the drink. Do you think some factors in the diet, in the background diet, could influence your results? I'm thinking about, for example, polyunsaturated fatty acids or something like that. If they differed between the study participants, would that potentially influence the gene expression?
Yeah, for sure. I think if the habits or of those individuals is different, if they are taking a lot of fibers or other groups taking a lot of cholesterol, this all can influence cholesterol absorption or the level of cholesterol absorption. So
Yes.
definitely the intake of fibers, the gut microbiota can also influence this cholesterol absorption. So if some people are taking a lot of fibers, they will also influence the cholesterol absorption. But we try to avoid this by giving them a standard meal and advise them to have a specific intervention that was intended.
Yes, thank you. I think you have highlighted several times in your thesis the importance to know who are the high cholesterol absorbers and who are the low cholesterol absorbers. And now in this thesis, you focus on the gene expression. And if I understand correctly, you anticipate that this kind of approach could be feasible or could be used in future as a better test for defining who are high cholesterol absorbers and who are low cholesterol absorbers. So if you were to plan this kind of tests, what qualities would you require from such a test?
Sorry, could you repeat the question because I couldn't understand the last part?
Yeah. Okay. Do you think you would at one day see this kind of a gene test as a, for example, in the healthcare, as a test that could define people who are high or low cholesterol absorbers, and therefore you could design the intervention, whether diet or other means? So is that the case that you anticipate? Yes. Okay, so if you would design such a test, what qualities, what requirements would you need for this kind of a test? I'm thinking of here, for example, the costs, because these tests surely at the moment are not cheap or easy to do. But what do you think in the future? What would you need from this kind of a test?
Yes, I think it's still a good approach to divide individuals as high and low absorbers. Then you can give the right medication for those patients. If you know that a specific group of people are high absorbers, you can then intervene with, for instance, with stanols as a dietary intervention or with ezetimibe as a drug intervention. And for those low absorbers, so they are high in endogenous cholesterol synthesis, you can give, for instance, statins and reduced rice for the nutrition intervention. And I would still think that is an important approach to follow to categorize those individuals. Because now the first line of medication is statins, and it is not always the good selection, I think, to lower LDL cholesterol. Some people will get many side effects, and they will not benefit that much from this medication or this treatment. And furthermore, a combination of treatment is also a possibility. So to combine statins with ezetimibe for those patients who can benefit from those two drugs. And you also mentioned that it's costly to do this study or but I think it is, I think we have to do it in this way. Then you are working with precision medicine.
Mm-hmm. Yeah, and maybe for the healthcare approach later on, one need to think about the costs and also the sensitivity and specificity of the tests and so on. And do you think you could have some other markers that maybe would?
Professor
Okay,
Lakina,
or is it?
Sorry, I have to interrupt.
Okay.
We have to proceed. We have two
Yes,
more.
that's fine.
And thank
Yes,
you
thank
for
you.
your time. And I would like to move on to Dr. McKay, and I hope he is available online because I don't see him. Dr. McKay, are you available?
I am.
Perfect. Then I would like to introduce you. Dr. McKay was also a member of your assessment committee. He's an assistant professor at the Department of Food and Human Nutritional Sciences. And for our guests here in the audience, he is now situated in Manitoba with his 7 hours time differences earlier. So it's for him middle in the night to address his questions. And I'd like to give you the word, Professor or Dr. McKay.
Thank you, Director, for that. I'd like to thank the candidate for the interesting thesis that I participated in reviewing, and I think there's quite a few interesting findings and expansions on the literature there. One of my first questions is regarding chapter 3 and 4. For this, you use data from 5 separate clinical trials that were completed over a period of time. Where I am located, we have lots of individuals who like to participate in the trials and often participate in multiple trials. Within those 5 trials, do you know if there were people who were members of multiple trials?
Esteemed opponent, thank you for your congratulations and your question. We have performed quality control for the data that we had of the 5 studies. We have considered a population structure. Unfortunately, we did not check for the relatedness. If we did so, then we were sure that we don't have a repetition or a relatedness in our cohort, and that will give more or better results to our output. We still did not publish this paper. We are considering this point, so we will check for that relatedness.
Okay, good, excellent. And so if you did publish, or if you didn't remove those individuals, what would be the impact of that potentially on your current outcomes?
I think the impact. So you can get now false positive association between the SNPs and the campisterol and citosterol, or the. But if we do so, then you remove the false positive associations.
Okay. The other thing that was done across those trials in slightly different ways was measuring the high absorbers and low absorbers using a couple of different techniques. How reliable is the measurement or categorization of someone as a low absorber or high absorber over time?
Yes, true. We have if you are asking them now about chapter 5, in categorizing those individuals as high and low, it is on that time slot that we did that. But I think normally it's more stable that if someone is a really high absorber, then he will be a high absorber for his life being. But this may change if the diet will change, or if the habits are changed, it can influence the absorption level of cholesterol for sure.
I was just wondering, because actually this was more again directed to chapter 3 and 4. Could you potentially look at the different time points, or have you looked at the different time points across all 5 studies? And are individuals that are low absorbers always low absorbers, or do they change time point to time point?
Actually, we did not check in chapter 3 and chapter 4 at the different time points if participant was changed as from high and low absorbers, but definitely this could be an option to check and see. Yeah, it's a good suggestion. Yeah.
If it's not published yet, just to get an idea of whether they're moving around, or looking at the baselines or the control intervention if they're always high or low absorbers. A follow-up to that, I guess, is if an individual is a high absorber of cholesterol, does that always mean they're low synthesizers?
Normally, yes, because it is a reciprocal relation. So if someone is a high absorber, then he would be a low absorber. Because the intestine and liver, they are working together. So there should be also a balance. So if there's a lot of absorption, then the synthesis of cholesterol will be lower. And if there's more synthesis, then cholesterol absorption will be lower.
So you're saying, I guess, in a healthy individual, or normally there's a reciprocal relationship, but could someone be a high absorber and a high synthesizer?
In my data now, what we see, I didn't see that, but I think it's possible. It could be that then you will have, if we assume that someone can absorb a lot of cholesterol and synthesize a lot of cholesterol, then I think he will have a very high total cholesterol. And I don't know this kind of phenomenon, but I assume,
Okay. From those studies in chapters 3 and 4, you have 400 and some individuals, so you could also potentially look at that. I guess, okay. Then in terms of, you were mentioning dietary interventions and pharmacological interventions that could be used to categorize these individuals. If you yourself, based on the current literature, were found to have an elevated cholesterol, LDL cholesterol, would you want to know if you were a high absorber or a low absorber to guide your treatment?
Yes, for myself, I think I would like to know if I am a high absorber, so I can take my precautions. And so those people who are high absorbers, they have also a higher risk to develop, for instance, cardiovascular disease. They can change their lifestyle. They can change their food that they are eating, and they can maintain a healthier lifestyle. So I think it's good to know in advance if you are a high or a low absorber. But it's a choice that someone can make.
And I guess just wondering, sort of, do you know from the literature, when people receive genetically guided lifestyle modifications, do they tend to be more effective than non-genetically guided lifestyle modifications?
If I understand correctly, if someone knows the effect of his genes on his health, how he will react.
Yes, so from randomized controlled trials, if you tell someone you should eat this diet because of the genetics versus you should eat this diet, are there differences in the number of people who make those changes?
I think that depends on the person itself. So we are different people, and each one, take his own decisions. Some people are wise, and I think they will, make the good choice. So they will go for a healthier lifestyle and so on. And some other maybe ignore it, or they want to not to take any steps to develop themselves. But I think that is more a human thing than,
And I guess the final question along that is, do you think the genetic information can help override that human nature of whether we follow healthy lifestyles or not? Because you said it's a very individual to the person whether they follow this. Most healthy lifestyle guidances are known. So does knowing your genetic risk make, is there a value, or do you think there could be value of that?
I think it's still valuable to make more research in this field and understand the genetic variation for the clinical side, for instance. Then they can still provide the right medication, and less cases maybe appear for cardiovascular disease or for other disease. So I assume, or I think, that the field of genetic is still, an opportunity to develop health.
Okay, thank you very much. I'll give my time back to Veronica.
Thank you very much. And the opposition will be followed by Dr. Van der Kallen. She was also a member of your assessment committee, and she's assistant professor of the Department of Internal Medicine. And I'd like to give her the word.
Sorry. Dear candidate, first I would like to congratulate you with your very nice thesis. Interesting work, and it expressed your knowledge on the field of genetics, expression data, but of course of cholesterol absorption. And I would like to congratulate your team as well. My question relates to the data presented in chapter 3 and 4. First, the question about the way you have analyzed the data. You used the 5 study samples, and of course, this gives a great opportunity to reuse the data. You have a larger study sample, so that's great that you have done this. But except that there are maybe disadvantages of this approach. So I would like to have your opinion about that, because you mentioned that it's healthy people, healthy adults. Some of them are a little bit, they displayed mild hypercholesterolemia. But if I look in the table, which shows the characteristics of your study population, I see some differences in age and BMI and gender. Do you think you have to have an extra adjustment in your analysis? Because now in the analysis, as I have seen in the materials and methods, you only adjust for study population and not for the separate characteristics. Can you elaborate on that?
Esteemed opponent, thank you for your nice words and your question. We have, true, if you look to the baseline characteristic, there was a significant difference in most parameters between the 5 studies, and except for gender. But we have corrected for study, and by this, we have corrected for everything. So we have corrected for the method, how the markers were measured. We corrected also for age, so for all parameters it was corrected. by using the adjustment study.
But if I have a more detailed look in the table, some of them are a little bit older, but do not necessarily, for example, have a high BMI. If I remember well, study population 5 was a little bit older, but the BMI was lower.
Yeah.
So it's not, if you just by study sample,
Yeah.
you can't correct for all, because it's not going together.
Yeah.
So you did. Do I understand it well? You did only adjust for study population.
True.
If you think about this right now, would you change your approach or not? Or would you?
We have only adjusted for study population, but still this possibility is open to adjust for instance, age.
Mm-hmm.
As age can influence the results. So people older in age, they may have also higher level of cholesterol absorption. Certainly, I will take it into account and adjust for it.
And I agree, you have to adjust for study population. So add that as well.
Okay,
So don't skip that one. I have a particular interest in gender differences. So would you suggest that you also add gender in your analysis or not?
So if I understand correctly, gender, so that I take into account the difference between males and females, and to correct for that as well. Yeah. Yes, we can also do gender-gene interaction, and we test for the difference. Is there a great difference between the female and the males? I expect to see some differences, because it's known that male and females can have different hormones, and these may also influence the level of cholesterol absorption.
Yes, I agree on that. Yeah, I have had a look in the literature, and indeed there are effect modifications.
Yeah.
So I think that would be great if you do that, because in the end, I think it will possibly have an effect on your interventions if you look further in your precision medicine.
Sure. Yeah.
That would be my idea. Okay, great. And then if you go to the results, as you showed in your presentation, there are a couple of genes associated with intestinal cholesterol absorption. Some of them are associated with the synthesis, and some of them are associated with LDL cholesterol. What do you think is one of them more important if it's related to health and disease, not specifically to cholesterol absorption, but for the individual?
No, I don't think that one is more important than the other. I think we should keep in mind that an individual have more than one gene, so a collection of the SNPs or a collection of genetic variation in the DNA is important to look into it. And upon this, you can decide more or have a greater picture from the output. So for instance, we can also use a polygenic risk score. The polygenic risk score looks into, it needs a higher sample size, a population around 1,000 or more. And then you test, for instance, for a specific disease, let's say for cardiovascular disease, and then you can say, okay, having this many SNPs and this different genes can increase the level of having cardiovascular disease. And that is the polygenic risk score, which could be a good indication for the risk of having a specific or certain disease.
And the reason that your study sample is too small, as you indicate, was the reason that you didn't use this genetic risk score?
Yes, true. Our sample consisted of 465 participants, which is too small to do this analysis.
Dear Ms. Candidate, would you like to finish your answer, or are you finished?
Yeah, I will finish that too. I still have 2 words. Yeah, so our population is very small to perform polygenic score, and maybe in the future we can redo this with a higher or a larger sample size.
Okay, thank you for your answers, and I will give back the word to the prorector.
Thank you very much. Dear Ms. Mokhtar, the time appointed for defending your thesis is now over, okay? The degree committee will now withdraw to discuss the quality of your thesis, but in particular the way you have defended your thesis this morning. I would like to request you and your company, your family, your friends over here to await the results of our deliberation and our return to this room. Thank you very much.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. The mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Yes.
Apologize. Dear Ms. Mokhtar, the degree committee here present has assessed not so much the quality of your thesis, because that was already approved, but in particular the way you have defended your thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor.
Thank you.
Professor Mensing is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take the floor with all the official registrations.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Fatma Bashir Abulgasem Mokhtar, the degree of doctor and grant you all rights attached by the custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and other members of the committee and affixed with the official seal of the university. Wel edel zeer geleerde Dr. Mokhtar, beste Fatma, dear Fatma. It is always a privilege that the promoter is the first who can congratulate the former PhD student with the newly acquired title. Also, I would like to congratulate Ibrahim, your parents, your sisters and brothers, friends, and of course, Elias. It goes without saying, these congratulations are also on behalf of Jochem. I know you were looking forward to this moment, not because you were looking forward to this short speech, but because the defense of your thesis was over. You were not really excited about that part of the PhD project, and I'm pretty sure that you would have preferred a Zoom meeting or even email if that was possible to defend your thesis. However, you survived, you did well, and I sincerely hope that it was not as awful as you thought this morning it was. I received an email at 6 o'clock in the morning from you. As you know, this is the time for a short overview of the last 4 years. You started in 2018. I did not really know you, but Jochem did. As part of your master thesis, you have examined in a hepatic cell line the impact of short-chain fatty acids on inflammatory pathways in relation to ApoA1 transcription. Jochem and Herman Popeyes were your supervisors, and they were really impressed how hard and accurate you worked and how well you interpreted the results. In fact, that project was part of the PhD thesis of Jihad and resulted in a nice paper that you co-authored. You started your PhD together with Maite, who defended her thesis a few weeks ago and is now on holidays in Mexico. In the beginning, we did not have a room close to ours. We were sitting on the ground floor, and you and Maite had a room at the first floor. I've been told that you did not really rely on your memory and had a small piece of paper next to your computer on which the name of Maite was written, just to be sure that you did not forget her name. Of course, the first floor was not the ideal location, and fortunately, you and Maite could move to the ground floor after a few months. There you shared a room with other PhD students, which made it easier to integrate with the group. However, it remained a challenge because your topic was a little bit out of scope, since you were not doing your own intervention studies like most of other PhD students, but had to rely on data collected earlier by others. It was up to you to reuse these data and to find so-called hidden messages in these data. The basic idea of your PhD project was to identify new genes involved in cholesterol absorption. It was a challenge. You not only had to identify new pathways involved in cholesterol absorption, but also had to pave many pathways to come to the results. Jochem and I were not really familiar with this approach, and I bet that there are not many YouTube videos that you have not seen to come to these results. We can only congratulate you with this perseverance. I even tried what the Arabic word for congratulations is. I had a wonderful patient teacher who was not really impressed by my pronunciation, but I will give it a try. Af Mabrouk.
Yeah, thank you.
Now, please do not answer in Arabic then, because then I'm really lost. Of course, we did receive some support for the analysis from other people, and are very grateful to Susanne Kort from the Department of Bioinformatics and Marcus Kleber from Germany for their help. It is really not that easy. It was really a challenge. I would also like to take the opportunity to thank the members of the Corona for being present and challenging Fatma with their questions. You have presented your results, among others, at the Maithamer meeting in Germany. Not something you really liked, I think, but you did very well. You got some, let me say, nasty questions, but you stayed calm and even laughed afterwards when you realized that the name of the questioner in Arabic meant monkey. In fact, you are not only calm from the outside, I think you are more calm than from the inside, but also very kind. Maybe sometimes a little bit too kind. As already said, you are also very social, precise and accurate. When you promise something, it is a guarantee that it will happen. Be proud of these characteristics, which are very much appreciated by all of us. Completing this thesis was an achievement, but certainly not the most wonderful thing that happened in the last 4 years. You and Ibrahim became the proud and happy parents of Elias, and we know you enjoyed every second of it. What will the future bring? You have not told me formally, but you cannot hide it anymore. A little brother or sister for Elias is expected, and this morning I learned it is a brother.
Yeah.
Congratulations again. In the last year, you and your family has also moved from an apartment to a house in Roermond, but I have the feeling that you would like to move a little bit up more to the north, to Eindhoven. Who knows what happens? Hopes all your wishes come true, and we would like you to wish all the best for the future. I would like to leave it with this short overview and wish you an unforgettable day, and would like to give the word back to the pro-rector. Thank you.
Thank you.
Thank you very much. Dear Dr. Mokhtar, how does that feel?
Yeah, very special, and I'm happy also with the nice congratulations. And
Perfect.
yeah.
I would like to congratulate you also on behalf of Maastricht University, of our Board of Deans, and I would like to extend my congratulations, of course, to your promotion team, who guided you in the last 4 or 5 years to get to this point. I noticed that there was some, it was not a straight road, but it was a road with some, let's say, some diversity, but I would always say it's the end point what takes the final goal. And you made a clear presentation. I would like to congratulate for this, even I understand it, yes, because it's not my field. It's more related to neuroscience. But, and your questions you received from all of the members were also well received, as I noticed before. I also would like to thank all the members of your Corona, particularly the ones who are online. I think Professor Leitine maybe left us already. And I admire Professor McKay, who is still in the middle of the night. And I would expect some sleepy eyes, but I think we see them later afterwards. I would like to thank also the members here of Maastricht University who prepared questions and who get involved in your opposition this morning. In my congratulations, of course, I go to your family here on the front line, and I was already puzzled who is being. I know your husband is called Ibrahim. Is that correct?
Yeah.
So I was thinking, who is Ibrahim? So on the right side, it's not possible, and I didn't expect it was the photographer. So that's nice. So congratulations. Have a great day. And I would also like to address the students in the back. They're always in the back. So we're looking forward to see your defense here in the future. It's basically a good example, and we're looking forward to your defenses as well. Saying this, I'm going to close officially this defense, and I wish you a beautiful day. It's your day, and enjoy it.
Thank you.
How are we going to proceed? First of all, there is organized there's organized a reception, and we're going to make here a picture in the aula. You don't have to wait for us, and I would already advise you, for those who are interested in some coffee, tea, I'm not sure if it's beer there, but something pie maybe is definitely there. You can already start and go to the rafter, which is on the right side of the entrance. We will follow in about 7-8 minutes, and we start first to have a picture here, including Dr. McKay, and then later we make another picture on the stairs of the hall of our aula. Thank you very much, and enjoy the day.
Thank you.